levels in genes related to cancer. So we can get a profile of transcriptional activity across the board to see which genes are associated with cancer. Finally, we can look at epigenetic modifications. That's also we talked about last week. And I mentioned that some cancer types are correlated with epigenetic reprogramming. Cancer is not a single disease, and this is the major challenge that we have in treatment of cancer. And that applies also to so-called same type, let's say breast cancer. It turns out that pretty much every patient is different, and the genetic constitution of cancerous tissues varies from person to person. So that's why personalized medicine or precision medicine approaches make sense, because we're trying to find drugs that are specific to a patient. So the goal is to tailor drugs towards specific cases, specific patients, rather than using some treatments that are supposed to kill active cells, which, as we know, have severe side effects across the board. So like I said, most cancer is sporadic, between 70 and 80%. Sporadic means that it's random. And we don't really see family associations. But there's some cancer types or cases for the same, even for the same type of cancer, we can see that some of them tend to cluster within families. So they form family clusters, and those cases account for between 15 and 20% of all cancer cases. And between 5 and 10% of cancer is hereditary, which means that there's some basic genetic mutation which is heritable, and we can identify the gene that causes this increased frequency of cancer. Those are the major 4 classes of cancer. And this classification is based on the tissue that various cancers originate from. So they can be classified into sarcomas, in which case the tumor originates in mesenchymal tissue such as bone, muscle, or connective tissue, or in nervous system tissue. Carcinomas is the second. They originate in epithelial tissue such as cells lining the intestine, bronchi, or mammary ducts. Hematopoietic or lymphoid, those are malignant neoplasms such as leukemias and lymphoma, which spread across the bone marrow, lymphatic system, and peripheral blood. and finally, teratomas, those are cancers of embryonic tissues that consist of several types of differentiated tissue. So those tend to occur early in development in infants or children. This is a general scheme for development of a carcinoma in an epithelial tissue such as colonic epithelium. And it shows you progression from normal epithelium to local proliferation with invasion across the lamina propria and then spread to local lymph nodes, and finally with distant metastasis to liver and lung. So tumors evolve from benign lesions first, and this is initiated by a mutation in so-called gatekeeping gene. In the case of chronic cancer, this often happens in this APC gene, which is a tumor suppressor gene, and we're gonna talk about that more in a moment. So then this mutation gives the cells some growth advantage. So they start to proliferate, they start to divide more efficiently than the neighboring cells. So they start to form a microscopic clone of cells that differ genetically with respect to this particular gene. And like I said, they form this benign tumor. And then a second gatekeeping mutation takes place in genes like this CRAS. CRAS is an oncogene, and we're gonna talk more about this gene today. And that gives these cells even more advantage, a selective advantage compared with neighboring cells. So they start to proliferate and they accumulate mutations in other genes related to cancer, but not only, so the mutation rate in general is increased in such cells because the repair mechanisms are less efficient, and the next mutations, when they happen, they occur in genes like this PIK3CA, which is a kinase, SMAD4, P53, which is a very important tumor suppressor gene, and this is when the gene invades first neighboring tissues and then starts to metastasize to other organs. So there were many federal initiatives. I started this lecture telling about Richard Nixon's initiative to fight cancer, but there were many more, and there is a whole NIH institute, National Cancer Institute, dedicated to research on cancer. There are great resources. One of the most impressive is this Cancer Genome Atlas, TCGA, which is devoted to the analysis of multiple samples obtained from more than 30 types of human cancers, from multiple patients, from hundreds of patients, in which case healthy and cancerous cells are obtained from and compared and analyzed. So that's a great resource. When someone is interested in the genetic changes that occur in specific cancer types. it's not only about mutations, it's also about gene expression, because turns out that cancer cells have some unique profiles of gene expression. And also, this is a way to identify genes that are associated with cancer. So the idea is that we can extract RNA populations from cancerous cells and non-cancerous cells and compare them with respect to expression of all genes. So we have now tools that give us access to these measurements. And when we have robust statistical replication, we can tell which genes are either downregulated or upregulated, which suggests some abnormal expression in cancer. So that's a great tool, that's a great clue to tell us whether or not specific genes are related to cancer, or if they directly cause cancer. So the first expression profiling, we don't have much time to talk about the methods, but you got a glimpse of those, I guess, in the biochem class. This is microarray, a method to profile gene expression. Nowadays, we tend to use sequencing, so we can sequence all RNAs. But at that time, which is like between 10 and 15 years back, the cheapest or the most efficient way to look at genome-wide expression patterns was this microarray method. So you can think of those individual samples as samples from the same patient. It could be like the healthy tissue and cancer, or it could be, or like in this case, we compare two types of cancer, tumor A and tumor B. It's just a hypothetical example. So each square like that with these cells represents a single sample of RNA. And these RNAs are labeled with some fluorescent factor, which reflects the level of expression or the quantity, and the quantity is translated directly into the level of expression. So let's assume that the green color when it's there reflects high expression or high quantity of a transcript, and red color is low or abnormal expression or below average. So those are individual samples, and then there's some computational work to do. So we reorganize the samples into this large panel. So now samples are represented. So this is like a reconfigured picture from the left side. So now samples are organized into columns, and every cell in this column and every cell in these squares here represents a single gene, right? But we have thousands of genes to analyze, and these genes are represented by rows. So every row is an individual gene. Every column is a sample. So with some extra computational work, now we can take some average values after some normalization, and we can look for differential expression. So those samples in tumor A in green have some overexpression compared with tumor B, or it can be, like I said, it can be any comparison. In most cases, we're interested in comparison of healthy tissues and cancerous tissues. to find some association with cancer. You have a question? This is, we say normal or abnormal relative to something. So like here, we see that there's a difference. This is upregulated, this is downregulated, but relative to each other, right? But if let's say this panel would be healthy tissue, then that would be kind of abnormal downregulation. I'm sorry, upregulation, because we have more of this transcript. So green color means higher level of transcription. So that's one way to find genes that are linked to cancer through their expression patterns. That make sense? So how much genetic heterogeneity is there among different cancers, or even between different metastatic lesions? In short, there is a lot. And it's not uncommon for a single metastatic lesion to have multiple 20 or more clonal genetic alterations that are not shared by other lesions. So first, this is intratumoral heterogeneity, which happens within the same tumor. So I told you that the tumor starts with a mutation in the gatekeeping gene, like first APC, and then the second mutation hits in another gene-related gene like CRAS. But just so these mutations lead to some microscopic or then macroscopic clones within a single tumor. So there is some clonal diversity, because you can, like, even when you think about these first mutations, sometimes there is an APC mutation in one cell and CRAS mutation in the same cell, but other cells may have only CRAS mutation. So those would be like cells that will have growth advantage compared with neighboring cells, but they will lead to this genetic diversity within a tumor. So this is reflected by these various colors in primary tumor. They differ genetically. They have, when you look at like mutational profiles, they differ in terms of mutations that they accumulate. And then this is a metastatic heterogeneity. So there's a metastasis, which is a process of migration. Some cells detach from the primary tumor and they acquire these migrational properties. So they land in other organs and they start to grow there. But you can think of the primary tumor as a complex of various, like a composition of various clones. So it's easy to envision that different clones from within the same primary tumor will lead to different metastatic lesions, which will start with different mutations, and then they start to accumulate, they start to diverge in genetic terms and accumulate some unique mutations. And there is this intrametastatic heterogeneity, which means that these metastatic lesions also grow and accumulate even more genetic variation there. And of course, there's an interpatient heterogeneity. Needless to say that there's huge differences between patients, even for the same cancer type. So that's the huge challenge to cancer treatment and finding cure, because there won't be a single cure to cancer, because every cancer is different. So we need to find solutions that are tailored towards specific cases, specific genetic mutations. So how many genes are we talking about? Turns out that there are at least 140 driver genes. By driver genes, I mean those that really are causative factors of cancer. So they really give cancer some advantage compared with non-cancer tissues. In typical solid tumors like colon, breast, brain, and so on, there's an average of 33 to 66 genes that display some somatic mutations. 95% of these genes are single base substitutions, and most of them result in missense changes. Missense changes are those that change amino acid sequence. There's some replacement, substitution at the amino acid level. But there are also some nonsense changes, those like premature stop codons or some other alterations, including splicing sites, UTRs, and so on. There might be also deletions and insertions of one or a few bases. Those are called indels, which includes both deletions and insertions. So I mentioned this term driver mutation. So now let's define them. Those are genes that underlie oncogenesis in a functional way. So they provide the growth advantage. And they occur in two types of genes, oncogenes and tumor suppressor genes. And it's a very important distinction that I'm going to spend some time on. But the major difference between these two is that oncogenes are characterized by a gain of function mutations. So they tend to have dominant effects. Compared with tumor suppressor genes that are characterized by loss of function mutations and therefore are mostly recessive mutations. But once those occur, the genome, the cancer genome becomes unstable, and the repair mechanisms are not as efficient as in healthy cells. So they start to accumulate, these cells and genomes start to accumulate multiple mutations that are not directly related to cancer. They're just side effects, so they don't really contribute to these cancer properties. They don't increase growth advantage. They, like I said, they just accumulate randomly. So they are mostly neutral. Neutral means that they don't contribute to the cancerous properties. So they kind of carry it along for a ride. That's why we call them passenger mutations. And like I said, this is the result of increased mutation rate in these cells. And insufficient genome repair mechanisms. So in general, the cancer genome becomes very unstable, and that also leads to some major reconfigurations, including chromosomes. That there are some abnormal chromosomal mutations happening in these cells. Here is another comparison between oncogenes and tumor suppressor genes. This is an analogy that I like. Because oncogenes are gain of function mutation, then and tumor suppressor genes are loss of function, then we can compare this to this racing car, in which mutations in proto-oncogenes or oncogenes would be corresponding to, so this is oncogene, would be corresponding to increased acceleration. So there is more gas because there is a normal function in a proto-oncogene, and then after the mutation there is more of the function. So it's there's some gain or increase in the function that is abnormal, and it leads to abnormal growth or cellular division in these cells. Compared with tumor suppressor genes, so tumor suppressor genes can be compared to brakes. So they put brakes on some genes. Once these brakes are gone, then the effect is the same. So there are mutations leading to cancer. There is this uncontrolled growth now, but the mechanism is different. It's because, again, there's no control, there's no inhibition conveyed through tumor suppressor genes. So that's why this analogy says no brakes. So it was very difficult to actually distinguish between TSGs or tumor suppressor genes and oncogenes. We knew what their effects were, but then when we started to obtain all these molecular profiles of cancer, including mutations, we noticed that actually mutational profiles of oncogenes and tumor suppressor genes are different. And when you think about it, that makes sense, right? Because in the case of oncogenes, like I said, these mutations tend to be dominant based on this gain of function effect, which means that the function is preserved, but it's just increased in its amount. So it's not easy to obtain such an effect, because there will be only specific sites that will lead to such effect. So this is reflected by these little triangles pointing to specific sites, to specific bases in this gene sequence. So the height or the number of these triangles reflects the number of mutations happening at this very specific site. So which means that this particular site is reoccurring in this PIK3CA kinase, which is an oncogene, and here's another example of an oncogene, IDH1. And this is this pattern contrast with what we observe in tumor suppressor genes. Because in tumor suppressor genes, you have a number of truncating and missense mutations that accumulate along the length. So there are no preferred sites. Why does it happen? It's because these mutations are inactivating mutations. These mutations kill the gene. So, in other words, it's very easy to kill a gene function compared with increasing the function, right? There are some very, just a few sites, a few mutations will lead to increased function, to gain a function effect versus inactivating missense or truncating mutation. Truncating means that there's this blue color means that basically the translation, there's some premature stop codon here, and the translation stops at this site. So even if the protein is produced, it will be truncated. It will be incomplete. And another striking difference is that you don't really see these truncating mutations in oncogenes, right? Because that would inactivate an oncogene. That would kill its function, not increase the function. Are you with me? So, in other words, based on the mutational profile alone, we can tell if a cancer-related gene is a oncogene or if it's a TSG. Which led to this 2020 rule, again, allowing us to differentiate between oncogenes and TSGs. So oncogenes have more than 20% of recurrent and missense mutations. And TSGs have more than 20% of inactivating mutations. And that pretty much applies to all known TSGs and oncogenes with no exception. So it's quite a useful rule. And it's sort of a new thing that we discovered looking at genomics of various types of cancer. To complicate things a little bit, you should know that there's some genes that with respect to whether they are oncogenes or TSGs, that's something that differs, that the same gene can be, actually may have some TSG properties or may have oncogenic properties dependent on the type of cancer. Because as you know, many genes have multiple functions, they play multiple roles, and the roles may differ from cancer to cancer. So that's some complication that kind of blurs this picture, but other than that, like I said, this rule is very useful. So I need to repeat what I said. So this is the difference. This is that we can't missense recurrent mutations and compare them with truncating or inactivating mutations. And so that's one thing. Another thing is that we look at the distribution along the sequence, right? So there are some in oncogenes. These missense mutations are recurrent, which means that we see them from case to case. It's the same mutation that happens in patients in multiple patients, right? Within the same, within cancerous lesions in the same patient, but also between different individuals, between patients. So that's why we call them recurrent, and you can tell that this is a recurrent mutation by this length of the stack of the triangle. So each triangle represents an individual sample. You can think of it as a person, as a patient, right? So now you see that all these patients had mutations exactly in the same position. And that's because these oncogenes, or proto-oncogenes, they become oncogenes through gain of function effect. So whatever function they had, and this function is related to let's say cellular division, that it's a signal for a cell to divide. So now the signal is preserved, but the cell produces more of the signal. So now the cells start to divide as crazy, right? Because this is the gain of function. This is the increased function of oncogenes. And like I said, it's easier to break the gene, to kill the gene, because you can inactivate it basically in any position of the gene. Any mutation that is like truncating, like a premature stop codon, or like it could be like an indel missing one letter or two letters that change the frame. They just inactivate the whole gene. They kill the function. So they may happen in pretty much any position along the sequence. So that's why the distribution is so flat. They are not recurrent. So again, every triangle like that reflects a single patient or a single case that we analyze. Okay. So now we just a simple score. We just count these mutations and look. We check if they're recurrent misses mutations, which means those spikes here, or if they are inactivating mutations that like truncate the protein product, which means those in blue. And again, the distribution is flat. They may occur pretty much in any position. So that led to this 20-20 rule. So if it's more than 20 of this type, this is oncogene. If it's more than 20% of inactivating mutations, then this is a TSG. If these ratios are not clear cut, it probably means that a gene plays both roles. It can be both oncogenic, dependent on the context, on the molecular, cellular context. It can function as an oncogene or TSG. This diagram shows the major cancer genes for some cancers, and the larger the gene name, the more frequently the gene is defective in that cancer type. So the arm with this BRAF gene corresponds to, or is supposed to indicate leukemias and lymphomas. This is breast cancer, this is lung, this is bone, liver, stomach, and intestine, and so on. So you see that some genes, and that applies to both oncogenes and TSGs, are recurrent, so they're really kind of promiscuous. They happen in many types of cancer, but there are some that are more unique to cancer. So those mutations are more useful from our point of view because they can be used as diagnostics for cancer. Or even before it really starts or it's dangerous. This is a diagram, or actually two diagrams, showing genetic mutations in lung adenocarcinoma and in squamous cell carcinoma. So you see that two different types of cancer, they differ in the mutational profile. So like in this lung cancer, like KRAS is the mutation that dominates in terms of its frequency. There's still many mutations that we don't know what they do. And this is a frequency of genomic alterations in squamous cell carcinoma. So you see that, for example, here, this kinase PIK3 amplification is the most dominant pattern, and it's also, when you combine it with these mutations, those are amplifications, which means the gene is duplicated, or mutations within the gene. So this is almost half of all mutations that half of the genomic profile of the cancer, which is well explained by this particular gene or oncogene. So what are oncogenes? These genes are not the genes that sit there just to cause cancer in our cells, right? These genes are very essential genes that play some basic cellular functions, and you can divide them into the major functions like cell survival, cell fate, and genome maintenance. This applies to DNA damage repair. And you have roughly 12 signaling pathways that regulate the three core cellular processes. And again, they include cell fate determination, like apoptosis, whether a cell dies or not, cell survival, and genome integrity or maintenance. I still have like 2 minutes to go over oncogene function, and I'm going to finish it tomorrow. This is oncogene function, and it also helps understand how they acquire these gain of function effects after they are mutated. So it turns out that proton congenes may encode growth factors. And so here's the growth factor, this little green cloud with L. So you can think of it as a ligand that binds to a receptor. So the mutations in these growth factor genes cause overproduction of growth factor. So there's more of this factor, and that increases the function. Proton congenes may encode growth factor receptors. So a mutant receptor no longer requires growth factor binding to support the function. So that again results in gain of function effect. Also, proton congenes may code for transcription factors. So those are proteins that bind to their target gene promoter regions, and they activate the gene. So there could be a mutation that changes affinity of this transcription factor. So these transcription factors now always bind to the target. They never disassociate from the target. And finally, proton cogenes may code proteins that transmit external stimuli for cell division. So there might be some mutations. After the mutations from proton cogene to oncogene, the stimulation is no longer required. So you can see that that all contributes to this hyperactive growth control pathway. And after these mutations, the growth is uncontrolled, increased compared to normal cells. Any questions? So we'll get back to it because this distinction between oncogenes and TSGs is very important, and it also relates to what we were talking about earlier in terms of what a dominant effect is, what the recessive effect is. So now you can see how that translates these general genetic terms, how they actually, what's their real meaning in conditions like cancer, okay? Thank you.